Okay, the first thing I want to point out before we really get started here is that this research was published online 2014, September 11th. Okay. Do many people get married on September 11th? I don't think so. I don't think people pick this date as a date for milestones in their life, for things that will be celebrated or remembered, important things that happen. I just think people usually try to avoid September 11th as a date for anything important, happy, milestones. I would think that having your research published on the NIH website is an important thing. And I just think that this date is suspect, okay? Point blank. I'm going to move on. I just wanted to point this out. I think it's strange that that's the date that they would do this, publish their work. Okay. Now, the research is by Selena E. Giuliano, Yale Stem Cell Center and Department of Cell Biology, Yale University School of Medicine. This name is tricky. I may get it wrong. Hyphen Lin, Yale Stem Cell Center and Department of Cell Biology, Yale University School of Medicine. And Robert E. Steele, Department of Biological Chemistry and the Developmental Biology Center, University of California, Irvine. Okay. Now, generation of transgenic Hydra by embryo microinjection. I am not going to read this out loud because this is supposed to be serious and not a comedy, but I will read certain parts. Hydra is used as a model for the study of regeneration, pattern formation, and stem cells. The development of transgenic methods has revitalized the study of Hydra biology. Generating transgenic Hydra is surprisingly simple and robust, and here we describe a protocol that can be easily implemented at a low cost. Okay. Okay, and let me go ahead and say that anyone who wants to read this as I'm going through it, because I'm not going to read the whole thing out loud, feel free to pause it and read it. I'm also going to link this in the post or in the description, depending on what site we're on. But yeah, if you want to just read it in this video, just pause it. So, Hydra has been used to study regeneration, pattern forming, and stem cells for approximately 250 years. Transgenic Hydra lines are established by injection of plasmid DNA into embryos, which results in random integration and chimeric expression in a substantial frequency of hatchlings. Okay, so I'm going to talk about a couple of things real quick. I'm going to talk about transgenic and chimeric. Okay. So transgenic. Genetically modified organism. Chimeric. Chimeras. Chimera is basically, okay, the definition, a fire-breathing she-monster in Greek mythology, having a lion's head, a goat's body, and a serpent's tail. Basically, a chimera is when they're mixing up different species to create new things. They're just throwing it in a bowl and cooking it up to see what they can come up with. I shouldn't even get started. I get all worked up with the chimeras. Anyway, so Okay. Transgenic is your genetically modified organisms. Chimeras are creatures that are made with multiple DNA, multiple species mixed up together, and a new thing is made. Okay. Let me jump over here real quick. Hydra are remarkable because they are immortal. Much of immortality can be ascribed to the asexual mode of reproduction by budding, which requires a tissue consisting of stem cells with continuous self-renewal capacity. Basically, a hydra, they don't need to mate, or you only need one hydra to produce another hydra. They just can self-replicate. So they're immortal. They can just create new hydra all by themselves. They don't need a partner. And they have the ability to fully regenerate. If a hydra is damaged, it can completely heal itself. If one hydra is cut in half, you get two hydra, because the part that's missing will regrow. So those things make hydra remarkable. All right, we're going to jump back over. Okay. So, a line with uniform expression in a particular lineage can be established by asexual propagation. Yeah. Okay, and in the 7 years since the first transgenic hydra lines were made, the reason I'm pointing out that sentence is because when we see 2014, we know to go ahead and take 7 years off of that to make it 2007, and that's when, assuming this is truthful, so 2007, because this was published in 2014, and it says in the 7 years since the first transgenic hydra lines were made. So that would have put it at 2007. And if I were going to bet on it, I would bet that the hydra left the water sometime around 2007. Hydra belong in the water. They do not belong on land. They do not belong on plants or trees, tree bark, tree leaves, tree stems, tree seeds, plant seeds, insects, people. They belong in the water. And I have a feeling that they probably left the water in 2007, or around 2007. Okay, so that's why I'm pointing that out. Now, again, I'm not going to read this whole article. I will link it. I was just pointing out those things. Took 10 minutes to point all that out. Good gosh. And like I said, if anybody wants to read it, just pause it and read it. I've scrolled through the whole thing here, and I will link it in the post. Okay. Hydra belong in the water. Hydra do not belong on the wood or in the wood. Hydra embryos do not belong on the wood. This is not right. Hydra belong in the water. Hydra do not belong on the leaves. Do you see them? Hydra belong in the water. They do not belong on the leaves or in the leaves or in the veins of the leaves. Hydra belong in the water. Hydra do not belong on the seeds or in the seeds. Let me just point out this piece of this Hydra sticking up with those little lights. Now imagine that right here in all of these little lines. And they do not belong inside the seeds. This is the seed pod, or the string that has the seeds on it from the hanging pin oak tree. I believe that's right. The seeds that I was just showing are from the sourwood plant. This is the hanging pin oak tree. Well, let's have a closer look at the hydras on these seeds. Not a single one of these is supposed to be here. They're supposed to be in the water. They are hydras. They're supposed to be in the water. They're not even hydras. They're man-made hydras. They're artificial hydras. They're not even legitimate hydras. But they're a problem. They're a very, very big problem. They are an enormous problem. They should have stayed in the water. Hydra belong in the water. Hydra do not belong in the dust. Dust is the opposite of water. Dry, dusty environments. Dust is not what you associate with wet. Hydra belong in the water. They do not belong in the dust. This and this. They are both Hydra. They're not a few. They're millions and they're billions. They're beyond count. They are beyond count. There's no number. They are out of the water. They are immortal. They can fully regenerate. They reproduce asexually. They don't need to. They can just make more. We have a problem. Hydra belong in the water. Hydra do not belong on the insects. Focus. Oh my God, this is hard. Wow, it's hard to do one-handed. We have